being in contact with all these great researchers that he also invited. It's also gives us a lot of learning. So Carl, he has a bachelor in physics from Oxford, and then he did his PhD in Cambridge, studying calcium signaling. And then his first postdoc was in the University of California, San Francisco, with Roger and Ecole, studying synaptic transmission and plasticity in the hippocampus. Then he did a second postdoc in the Max Planck of Heidelberg with Bert Zachman. And when he began working on primary somatosensory barrel cortex, investigating cortical circuits and sensory processing. Then he went to Lausanne, and he joined the Brain-Mind Institute at the EPFL, that is the Polytechnique Federale de Lausanne, and where he started a lab of sensory processing. And so today he's going to show his work, and he investigates the functional operation of neurocircuits in awake mice during quantified behavior. Thank you very much for accepting the invitation, Carl.
Great. Super. So thank you very much for the kind invitation. It's a great pleasure to be here. I've had a lovely day, so it's been very interesting. So today I want to tell you a bit sort of about our current work, where we're sort of been interested in sensorimotor transformations. We sort of come mainly from the idea of sensory perception, and more, I think, maybe perception is a difficult word to deal with, and sensorimotor loops are easier to simply discuss. But nonetheless, I'll sort of start by introducing our main interest from the idea of sensory perception, where I think we're all aware that sensory percepts are not out there in the world, that they're actively constructed by the nervous system. And so we can have images like this, where there's one set of information that falls on the eye, and yet two different percepts can emerge, that we either see the black cup or we see the two faces looking at each other. And so clearly, one set of information falls on the retina, and the brain then alternates in that sort of the subjective percept as to what we actually think we're looking at. And so the goal of sort of my lab then is to try and understand causal mechanisms for sensory perception. And we'd like to know that at the level of individual neurons and their synaptic connectivity, and try to understand how sort of causal and mechanistic function of synaptic circuits are involved in generating sensory percepts. And of course, as soon as we start talking about sensory percepts, we need to have a behavioral report. It's a subjective thing, the sensory perception, and so we need to have a motor output from the animal. And so a key thing, obviously, is for animals to tell us what they think they're feeling. And so we've been looking at this in mice, and so this is sort of highly simplified, sort of, I think, the simplest possible behavior that one could think of, a detection task, where we have head-restrained mice, so we put little metal head posts there. We teach the animals to sit still under head fixation. We then furthermore teach the animals to drink from a little spout so that they know that they can get water reward if they lick. And then in sort of the interesting part of the training, then we make water delivery contingent upon a whisker stimulus. So the animal sitting here on top of an electromagnetic coil. We put a small piece of metal on the whisker, and then at random times with sort of a mean interstimulus interval, something like 8 seconds, we then give a small 1 millisecond electromagnetic pulse that then sort of pulls the whisker down with a 1 millisecond of force, and it comes at a random time, so the animal's waiting, and eventually there's a little doing on the whisker. The animal has to perceive that, realize that that predicts water availability from the spout, and then if it licks, then it gets a water reward. And of course, if it doesn't lick, it gets nothing. And if it licks at other times, it also gets nothing. And if anything, it gets a timeout and a delay. So we then have different trial types. We stimulate, the animal licks, it gets rewarded, and so this we call hit trials. We stimulate if the animal doesn't lick, of course it's unrewarded, and we can think of those as mistrials. And there are also, of course, trials where we don't stimulate, and if the animal licks, we might think of those as false alarms, and if it doesn't lick, we might consider that a correct rejection. And so that's sort of the basic paradigm that we're interested in, and we think that although it's an extremely simple behavior, it has at least sort of some of the core features of sensory perception, that there's a relatively arbitrary sensorimotor transformation. We put in this meaningless stimulus for the animal, just a doing on the whisker, and it has to translate that into a meaningful goal-directed action in order to obtain a reward. So we've looked a little bit sort of at the learning process, and sort of the long-term goal that we have is to try and understand the learning mechanisms of how you take and sort of an arbitrary sensory stimulus and make something useful out of that. And so that's sort of the real long-term goal, is learning to, as it were, perceive or to at least translate sensory information into something useful. But I'd say we haven't approached the learning question yet, so I'm going to simply tell you sort of about what happens at end stage after animals have learned. But this is sort of the learning curve. So on day one, they go and they know that licking is a good thing, and so they're licking a lot. But they lick just as much on the stimulus trial. So these are the trials. This is the hit rate. So this is the fraction of stimulus trials where they lick. And this is then the fraction of trials when we don't stimulate that they lick. And on day one, it's the same. They have no idea what the task is. We stimulate, and they don't know that that stimulus is something that should be meaningful and direct them to licking. And so gradually, day by day, so this is training day, they go in for sort of 1-hour training sessions each day. And over time, over a period of about a week, they learn that licking after the stimulus is good, and they get rewarded. And if they lick, they learn to suppress licking when there's no stimulus. So sort of it's the divergence then of these two curves that tells us that learning takes place. And as such, we would love to understand this learning process of how, through presumably trial and error experience, the animal learns that it's a good thing to lick immediately after a whisker stimulation. But right now, what we do is we train the animals to some sort of criterion around 80% hit rates, less than 20% false alarms, and then we simply record here once the animals are, as it were, experts, or at least have reached some sort of stable criterion in their performance. We've also looked a little bit at the psychophysics of the process, and of course, as you turn the stimulus strength down, they lick less. Presumably there's detection problems at the sensory end, where sort of small stimuli in the end merge, presumably with noise, and they don't lick more than the false alarm rate. Again, sort of disappointingly at this stage, so far we've simply analyzed just the simplest point, so where we make big stimuli. So these are stimuli that presumably don't suffer from sensory problems at the periphery, that presumably there's a good translation of the sensory information through to some station of the brain, and it's the translation of that sensory into a motor output that's a failure. So we sort of get, even for the largest stimuli, we end up with something like 80% hit rates, and so there's still 20% of the time or so that they miss. And what we're going to study here is largely what's happening in that hit versus miss condition. So we're not sort of at the level here where we're sort of dealing with detection thresholds, and that's why I think probably what we should think about in terms of this data is that it's really a sensorimotor loop that we're studying, and that somewhere along the line that sensorimotor transformation fails in the miss trials, whereas of course in the hit trials there's a completion of that sensorimotor loop and the animal ends up licking. So for the whisker system, there are a few nice features of it. So if you stimulate a whisker, there are mechanogated ion channels that presumably are sitting here on the sensory nerve, the trigeminal nerve. You move a whisker, and that initiates action potentials down the trigeminal nerve. First glutamatergic synapse in the brainstem, second glutamatergic synapse in the thalamus, and then the information's further processed here in the primary somatosensory cortex. And one of the nice features here is the timing, that if you deflect one of these whiskers, it's about 5 milliseconds or so until you see information processing in primary somatosensory cortex. So we think that sort of tight temporal correlation might be helpful in the long run in terms of understanding the information flow. The other feature that's nice about the system is that there's a very nice one-to-one mapping of the whiskers that's in an anatomical map. So this is the work of Van der Loos and Woolsey in 1970. They discovered that there was a one-to-one mapping of the layout of the whiskers on the periphery and the somatotopic layout in primary somatosensory cortex. So there's sort of large-scale, very obvious anatomical correlates of each individual whisker here in the form of what's called a barrel. And so we like to put sensory information on one particular channel, the C2 whisker. That's where we put all the little 1 millisecond pulses. They come in here, and there's then a sort of an anatomical location here of the C2 barrel column, which is nice in the sense that this is about 200 by 300 microns, so it's a relatively small, well-localized region that we can see anatomically, and we think that that's some sort of, in the long run, will be important because this hopefully will allow us to make more detailed correlations between structure and function. On the other hand, it turns out that in vivo, if you look at the brain, you don't see these barrels, you simply see the surface blood vessels, and at this stage we don't have ways of looking into the brain and simply identifying where the C2 column is in vivo. Of course, post hoc, we see the nice barrel pattern, but that's only post hoc. And so our experiments start with intrinsic optical imaging, a very simple technique that Amiram Grunwald developed many years ago. We shine red light on the surface of the brain through the intact skull. We deflect the C2 whisker, and we then localize that C2 whisker. In this case, we drill the craniotomy afterwards, put some red dye on the surface, cut tangential sections, and here you see sort of the reality of the barrel map inside layer 4, where you see these cell-dense walls that then surround each one of these sort of barrel columns. And of course, as one would expect, there's a nice one-to-one mapping of the functional location and the anatomical location of the C2 whisker. Now, what's useful about the intrinsic imaging is not so much that we can find out where the anatomy is, but this then allows us functionally then to put in viruses, drugs, or recording electrodes and target it specifically to the main whisker representation of the C2 whisker. And so the first experiment that we then sort of do here in the detection task is to try and see whether this area of the brain is actually involved in the detection task. You could sort of imagine that this is extremely simple. We stimulate a whisker, the animal licks. The whole thing could be accomplished in the brainstem without any involvement of neocortex. And so this is then sort of the first experiments we did, is simply to inject tetrodotoxin to block voltage-gated sodium channels, block action potential firing in the cortex. And so the animal sitting in head fix is performing the task. So we're stimulating at these random times, about 80% hit rates, and at some unknown time to the animal, we inject tetrodotoxin, and we get a rapid decrement in the hit rate of this task. And so apparently, action potential firing in S1 barrel cortex is important for performing this task. We don't know how far the TTX is spreading in this case. The only sort of spatial information we have at this time is we inject into forepaw primary somatosensory cortex. It's about 1.5 millimeters away, and that has no impact upon performance. So these are some spatial localization to the inactivation data here. And similarly, we can block glutamatergic synaptic transmission, block AMPA receptors or NMDA receptors, and we see a similar phenomenon. We see a block of the, or at least a reduction in the hit rate. And so apparently, glutamatergic synaptic transmission and action potential firing appear to be important for execution here.
Yes? Each individually, both together?
So we've only done them together. We haven't tried individually, but I think that would, of course, be an interesting experiment to do, but we haven't done that. We've also done the Converse experiment, where we stimulate and try to do substitution experiments. So in this case, we express channel rhodopsin. So again, we inject an AV vector into the C2 barrel column. Again, spatial specificity is not as high as one would want it to be, so there's some spread of the virus. So in the end, we're infecting neurons that are probably extending across some 500 microns or so multiple cortical columns. We express specifically in excitatory neurons, as EMX1 Cree mouse, an AV flex vector, and the expression time is something like 3 weeks or so until it stabilizes. And so during that 3 weeks of expression time, we then train the animals to perform the same task as before. So we simply just put the metal particle there. We train the animal to perform the detection task. It never sees any blue light stimuli until a transfer test day, and on that day, we then start dropping in 5 millisecond blue light flashes. And what we were happy about was that in the majority of the animals that we tested, the very first 5 millisecond blue light flash, the animal licks. And there seems then to be a relatively rapid transfer from the animal's perspective in terms of a subjective percept, or at least in terms of the licking behavior of the animal, that the whisker stimulation and cortical S1 stimulation are equivalently interpreted by downstream motor centers. We can also check the other way around.
These channel rhodopsin is for all cell types, or only?
It's excitatory cells, yeah. So it's only excitatory cells. So it's EMX1 Cree for the aficionados. That's cortex-specific excitatory neurons that express in this mouse line. So we can also do the Converse experiment, where we first teach the animal to lick in response to cortical stimulation, so it's the same virus manipulation. In this case, we just stimulate with blue light. The animal learns to lick in response to that blue light stimulation, and then on a test day, we then give the very first whisker stimuli to the animal, and again, we find out that there's quite a nice substitution. So we can sort of go either way. We can teach the animal on the whisker, and that then transfers to cortex, or we can teach the animal to respond to cortical stimulation, and that then transfers to the real whisker stimulation at the periphery. And the reason we think this works so easily is because of the enormous simplicity of the task. It's sort of a dawing on a whisker, and it's a dawing on the cortex. And presumably, if we were looking at more complex things, we would have a much harder time getting any of this to work. And so the simplicity of the task is at one level, of course, a huge disadvantage. It's a very uninteresting task. On the other hand, it allows us to do simple things and perhaps draw simple conclusions about what brain areas at least are involved. And so we have sort of first order and conclusions that we think that S1 cortex is participating even in this extremely simple task. We think that there's both action potential firing and glutamatergic synaptic transmission that are required in S1 barrel cortex. That's the pharmacological experiments. And we can also do these substitution experiments where stimulation of S1 will replace whisker stimulation in trained mice, or we can train mice for the S1 stimulation, and that then transfers to whisker stimulation. Yes?
There's no delay between the stimulus and the time that the animal needs to respond to the lick?
So we have so it varied a little bit during the experiment, but at this stage now we have fixed 1 second reward window. So we stimulate, and the animal has 1 second in which it can lick. So and typical reaction times are sitting around 400 milliseconds.
There's been this ongoing debate about whether the barrel cortex is crucial for different kinds of behavioral things, and what different kinds of conditioning traits and delays. How does this fit into that?
Yeah. So I think that's an interesting question. So I think here, my guess is that there may be a need for this sensory information to be maintained a little bit. I mean, the stimulus at the periphery is 1 millisecond in duration. And so it may be that you do need to have some element of, I don't know, short-term memory, or at least some trace of that is maintained for some period of time before the animal realizes, okay, that's a whisker stimulation, I need to do something about it. And so I think this sort of fits into some sort of maybe intermediate sort of location. So it may be that if we'd done sort of stimulation for 1 second and the animal was allowed to respond within that 1 second, maybe then cortex wouldn't be involved. So it's not like we've tried exploring a whole lot of different variables here. And so that's the one thing we started on. And in all honesty, I was surprised that cortex appears to be involved. So for me, I thought maybe in the learning of the task it might be involved, but not in execution. But so far it looks like execution. It seems to be an obligatory node in the process. Yeah.
If the barrel itself and its whisker are incongruent, aren't the same ones, is there any transfer? I know that the problem is the thread of the optic.
Yeah. Yeah.
If you go far enough.
So I think that's okay. So again, maybe I should have mentioned the sort of the control experiment we've done here is again stimulating the 4-pole representation. There's no licking response to that. But okay, that's of course quite far away. My guess is that there's going to be transfer to nearby whiskers, and there certainly is in some animals, and sort of we haven't got very deep into this, mainly because we see just a huge amount of variability. So some animals, you stimulate nearby whiskers, and that seems to be sort of almost as good as stimulating the main whisker you've trained to. And other animals, there seems to be more specificity. And at this stage, we don't understand so much about sort of this sort of generalization or other processes. But I think that's an interesting thing that we'd like to look at in the future. But what we're mainly seeing there is variability, and so we haven't sort of got very far in exploring that at this time. So this then draws some attention to us for what's going on in S1 cortex during the detection task. And over the years, we've been studying layer 2-free neocortex in at least some level of detail. And we've made patch clamp recordings from identified neurons. So this is a layer 2-free pyramidal neuron, and we can record the membrane potential. This is from an animal that's not performing any tasks. But just to sort of give you the flavor of the type of data that we're gathering, where you see membrane potential trajectories on a variety of timescales. So there are slow fluctuations. This is 1 second, so you can see that there are sort of interesting timescales in the fluctuations there, and these are huge in amplitude. There's 20 millivolts, so the fluctuations are large in amplitude, and Some of them are taking place on slow time scales, and then you can have equally large changes in membrane potential that are occurring on much shorter time scale. So there's sort of a whole variety in the dynamics of the membrane potential, and of course, every now and then you cross thresholds and a spike is emitted. Yes?
Was the animal whisking during?
This is without whisking. This is just an animal that's sitting there, it's awake, but it's not doing anything under these particular conditions here. And of course, these membrane potential fluctuations we think are largely being driven by glutamatergic EPSPs and GABAergic IPSPs that are coming in across the somatodendritic axis. And of course, the individual unitary PSPs on average are small. They're about a millivolt or so and 10 milliseconds or so in duration. And so presumably, this is a result of network activity that's driving the membrane potential up and down. And the synaptic computation then is then to emit the spike or not. And the AP threshold seems to be a relatively well-defined number that's relatively stable for an individual neuron. So there's at least something relatively simple here. Now this is in animals that are not performing tasks, and of course an interesting question is how does this appear in animals that are performing tasks? So I'm just going to show you a couple of trials here. We stimulate the whisker, and you see an immediate early depolarization. So there's a sensory response. We expect that. It's primary somatosensory cortex. And that early sensory response is there in hit trials and miss trials. So this is a hit trial. We stimulate. There's licking of the animal. The reward valve is opened, and the animal enjoys some water here. We stimulate. In this case, the animal doesn't lick, and there's no reward, of course, delivered. In both of these cases, there's an early sensory response. And if you go across trials for this particular cell, you'll see that the early sensory response is invariant to the sort of the percept of the animal as reported by licking. So there's a relatively reliable stimulus. You stimulate, and the cortex responds equally on a hit or on a miss trial. And so that's good. This is primary somatosensory cortex, and it's sort of nice if there's a reliable representation of the periphery.
Which layer?
This is all layer 2 free. So everything I'm going to show you is layer 2 free, and it could, of course, be that things are very different in other layers. But at this stage, our recordings are all layer 2 free, and we're only just beginning to look in deeper layers at this time, but we don't have any data for the detection task yet on that. At later times, something more interesting seems to happen. So here, hundreds of milliseconds before licking, in this hit trial, you see that there's a depolarization, and there's even a couple of spikes being fired. The miss trial, there's also some depolarization, but it appears to be less than the hit trial. And that's indeed true across the sort of the average for this particular cell, somewhere 100 milliseconds or so after stimulus. There's a divergence in the membrane potential traces. The hit trial is depolarized. The miss trial is a little bit more hyperpolarized, and there's more spikes being emitted on the hit trial. So there seems to be two components to the sensory response. There's a reliable component that's being presumably driven by the thalamocortical input that comes in and appears to be invariant to the percept of the animal. And then there's a late component that correlates with the percept of the animal as reported by licking. Yes?
So with the channel rhodopsin experiment that you were describing a couple slides ago, if you increase the light intensity or the duration, does the miss rate go down?
So we looked a little bit, I wouldn't say in huge detail. What we did was we varied the duration of the light applied. So this is now in the experiment where we've taught the animal to respond to a whisker stimulation, and then we look at the substitution. And so the first stimulus we give is always 5 milliseconds, and then we varied 1, 2, 5, 10 milliseconds, and there's some dependence. So 1 millisecond, there's no substitution. 2 milliseconds, it's a little bit. 5 milliseconds is already pretty good, and 10 milliseconds is not much better than 5. So there is some dependence upon the stimulus intensity. So this is a single cell. This is group population data, and you'll see that sort of the effect is less impressive, but nonetheless sort of similar. There's an early sensory response that's invariant comparing hit versus miss trials. About 50 milliseconds or so after stimulus, the two membrane potential traces diverge. Hit is depolarized compared to miss. It's a small effect on average across the cells, but turns out to be statistically significant. In terms of spike rates, we see something relatively similar. The first peak in the firing rate here is the same, so there's no statistically significant difference between hit and miss trials in the first 50 milliseconds. And then as you look in the 50 millisecond bin and onwards, you see that there are more spikes on the hit trials than on the miss trials, and that turns out to be a sort of a factor 2 when quantified in this 50 to 400 millisecond range. And so some of those spikes then might contribute to driving ultimately the motor response of the animal. So we think that this sort of late depolarization then is interesting. It seems to be sort of a correlate of the subjective percept of the animal, and we think that there are related things, or there could be related results in human recordings. So this is from the work of Stanislas Dehaene, where they do EEG recordings. They give different visual stimuli to the human subject, and they have some masking here that then alters the percept of the visibility of the stimulus. And the early sensory response here in the sort of EEG accorded sort of here across the early visual areas is very similar. So there's an early response that seems to be reliable, and then there are later components, several hundred milliseconds later, where there's a late component that correlates with perceived stimuli and less in not perceived stimuli. And that then looks a bit like what we see here in the mouse somatosensory cortex. And so there could be some sort of more general importance to these late depolarizations than just in this whisker detection task. Of course, one thing that we don't know from human studies is whether this is in any way causally related to the percept of the subject. And this is something that we can investigate in mice. And so we then wanted to be able to do inactivation experiments with high temporal precision. And what we chose to do then is to express channel rhodopsin in parvalbumin expressing GABAergic neurons. So these are the fast spiking GABAergic neurons that tend to sort of give perisomatic inhibition, and we think these are amongst the more powerful of the GABAergic neurons. And so we have PV-CRE mice. These are the mice made by Sylvia Arber many years ago, and then we inject the same AAV flex channel rhodopsin vector. We express channel rhodopsin in the neocortex, and then by turning the light on, then we can then get these cells to fire and release GABA and suppress the nearby excitatory neurons. And so here's sort of an example experiment here. We're recording the membrane potential of a neuron. Here we give our C2 whisker stimulation. We see the sensory response coming in. And the first thing we did was to do early light applications here at the same time as a whisker stimulus occurs. And then when we do that, we, of course, take out the sensory response. So we hyperpolarize very rapidly. The sensory response is gone. And if we now look at the performance of the animal, we see that there's a deficit in the licking of the animal. So apparently there's a deficit in the percept, and that's not surprising. We've taken out the sensory response, and if we think that S1 barrel cortex is involved in the task, as we sort of thought from the pharmacological experiments, then this is entirely expected. So this sort of provides further confirmation that S1 is really involved in task performance, and it's involved at that specific moment in time when the stimulus is being driven. So it could otherwise, of course, simply be changes, downstream changes in brain states and other things when you do pharmacological inactivation. Of course, you don't know. You may know something about the localization of your drug, but you don't know about how many other targets are being affected by these experiments. The real thing that we were interested in, of course, was to see what happens during the late component if we turn light on 100 milliseconds or so after the whisker stimulation, and we hyperpolarize and we take out late depolarizations in the cortex. Does that affect perception? And here we again see that there are significant deficits in the licking of the animal. So presumably the percept of the animal has been worsened. It's a smaller effect than we see with the early component, and so and neither of these are as complete as a pharmacological inactivation. So we don't yet know whether this is because the optogenetic inactivation is less efficient, or whether this indicates that there are actually multiple different parallel pathways that are involved in task execution. So from this, we then sort of think that the late depolarization correlates with the percept of the animal and also appears to contribute to actually driving at least the motor response of the animal. Yes?
Is it possible to teach the animal to simply respond to depolarization, activation of the GABAergic neurons?
So we haven't tried. Certainly under our conditions, they don't. So, I mean, if you sort of teach the animal in the whisker task and you just start stimulating the GABAergic neurons, they don't respond to this. But probably one could teach them. My guess is you can teach them to do many different things. We haven't tried that, but I would guess it's possible, but we haven't tried. So here we sort of artificially stimulate the GABAergic neurons and see that we can cause deficits in sensory percepts. We also actually think that this is what's happening on a sort of trial-by-trial basis in terms of the activity of the PV neuron. So here we're now targeting extracellular recording, so we're looking at the spiking of parvalbumin neurons. And here is a cell recorded during a HIT trial. We stimulate. The PV neurons are extremely responsive, so they're the ones that respond with most spikes in the neocortex to a stimulus. So there's a lot of responses early on. But then on a HIT trial, there are less responses late with preceding licking compared to a mistrial where there's more action potential firing. Again, these are just two individual trials, but this is the data, group data from 13 different cells. So this is the data set that we have at this time. We stimulate. The early response to the parvalbumin GABAergic neurons is invariant comparing hit and mistrials, but then after that first 50 milliseconds, there's a divergence in the spiking of these cells, and on mistrials, the parvalbumin cells are firing more spikes than they are on hit trials, where we sort of return to baseline levels more rapidly. And so it looks like this modulation of the PV firing is sort of reciprocal to the excitatory cell. So on a hit trial, the PV neurons turn off, and that then might allow the excitatory neurons to fire action potentials, whereas on a mistrial, the PV cells are firing at higher rates, and that then might contribute to suppressing the excitatory cells. And so we think that the PV neurons are then involved in a gating process of allowing the neural circuits to sort of get a sensory percept and translate that into a motor action. So this is sort of an unusual way for the GABAergic neurons, the PV GABAergic neurons, to be firing. Usually, if the excitatory neurons are firing a lot, then you expect the PV neurons to be following. So this is the strongest synaptic connection in the brain from excitatory neurons to PV neurons. And here we're seeing the reverse situation. Here the excitatory neurons are firing more, but the PV neurons are firing less. And so there's something interesting going on here where it looks like the PV neurons are actually in control. They suppress their firing rates, and they allow the excitatory neurons to fire action potential. So this is an unusual situation as far as we're concerned. And of course, we'd love to know what's going on with the regulation of the PV neurons. But at this stage, we don't have any good clues. We've looked at the other subtypes of GABAergic neurons, and there doesn't seem to be anything particularly interesting going on in VIP neurons or somatostatin neurons during this period. So it seems to be the PV neurons somehow are having a different correlation with excitatory neurons than the normal under these particular conditions. And that's then sort of something that we want to investigate in the future. So the sort of the intermediate conclusion here then from sort of S1 cortex is that we have two sort of response parts. There's an early response that we think reliably encodes the sensory stimulus, and there's then a late depolarization that correlates and we think also contributes to driving the behavioral report of the sensory percept. And we think the PV neurons are somewhere involved there in their gating process of allowing the excitatory neurons to develop an activity that's appropriate for driving the motor output. So we think, sort of on the basis of this data, that we think S1 is somehow critically involved in this detection task, and we think that S1 has a motor role. So if you stimulate S1 motor cortex, it makes a difference to whisker movements, and there's actually a relatively direct pathway from S1 down to brainstem, premotor neurons, and so there's a direct motor action of primary somatosensory cortex, but we don't think that's how the animal is licking. We assume that there are other intermediate areas in the brain that are involved in interpreting the signals for S1 and converting that into licking behavior. And so sort of for the rest of this talk, I'd like to tell you a little bit about what we've sort of been investigating in downstream targets of S1 and how they might be sort of differentially activated. And so the first sort of little story I want to tell you about is voltage sensor dye imaging during the detection test. This is work by Alexandros Kyriakitos, a talented postdoc in the lab. And so we've been using the technique that Miriam Grünwald developed many years ago. We use one of his dyes, RH1691. We applied topically to the surface of the brain, it diffuses into the brain and labels sort of the upper half of the neocortex under our conditions. And this dye it's a lipophilic dye, it sits on the plasma membrane, and it changes its fluorescence depending upon the electric field across the plasma membrane. And if we make patch clamp recordings from a neuron that's sitting somewhere around here, and we compare it to the fluorescence that we record from a sort of a relatively large area, we find that there's really very close correlations in terms of the membrane potential of a single cell and the voltage sensitized fluorescence that's presumably resulting from thousands of cells. And there's sort of quite a surprising correlation there where a single neuron seems to predict relatively well the population behavior. And we think that results from very strong correlations in the membrane potential fluctuations of most neurons. You take any two neurons, you record their membrane potential, and the first thing you see is huge correlations in their fluctuations. And we think that forms the basis of the voltage sensitized signal. We're looking at the correlated membrane potential fluctuations in that given area. So we looked at this then, of course, during the detection task. And the first thing I just want to sort of point out is that if you sort of focus your region of interest on the primary somatosensory cortex and you do the detection task experiment, we stimulate, we see, again, two components. We see an early sensory response that appears to be reliable comparing hit versus mistrial, so there's an invariant sensory response. And then some 50, 100 milliseconds later, the fluorescence traces diverge, the hit trial is depolarized, so it's increased fluorescence, and the mistrial sort of gradually decays off back to baseline. And again, all of this occurs hundreds of milliseconds before the first lick of the animal. So this is all sort of somewhere between stimulus and response of the animal. You see that divergence of hit versus mistrials. So this is sort of nice. We have a second independent verification of the membrane potential recordings that we did with the patch electrode. Fluorescence gives us something similar. But of course, the nice thing about VST imaging is not that we can look again at S1 cortex, but that we can look over much larger brain areas and see what's going on. And so here we now look at average trials for a single animal. So this is hit versus mistrials. And soon after whisker deflection, we have sort of localized sensory response that sits, of course, in the sort of somatotopic location of the whisker that we stimulate. This, over the next milliseconds, begins to spread, so it now encompasses the last part of the primary somatosensory cortex. And in addition, it activates an area here in frontal cortex that we call the whisker motor cortex. And both of those components seem to be equivalent between hit and mistrials. All this in the first 50 milliseconds again, there's no difference. We get that sensory to motor response, and it doesn't seem to be strongly dependent upon the report of the animal. If we then look a little bit later on, we see that there are, of course, very big differences comparing hit versus mistrials, and these relatively widespread depolarization across many brain areas that are occurring on hit trials compared to the mistrials. And so sort of before, what we were looking at is what's happening in S1. So if you look at S1 over here in these late phases of the hit trial, you see that it has got warmer colors here than it does here. So that's what we're sort of looking at. There is that depolarization in S1. On the other hand, it's not the spot that you would choose to look at. If you sort of now start sort of from a different point of view, you might be more tempted to look sort of at frontal areas or maybe this parietal association area that seems to be more strongly differentiated between hit and mistrials. So we've now done this in 16 mice, and here I show you the superimposed data we align now onto the S1 C2 barrel column. And again, so this sort of averages of averages, where we try to sort of simply see what's the overall picture, what are the most interesting areas in the dorsal neocortex to look at in terms of comparing hit versus mistrials. And so you'll see that there's sort of, in these images, it'll loop around. You'll see there's the early sensory response, there's the early response in motor cortex. You'll see this late depolarization that's here across the parietal area. And I think you'll also notice that there's an interesting frontal spot here that's not the same as the whisker motor cortex. It's sitting about 1 millimeter anterior to it. And we think that this is maybe an M2 area, sort of that's sitting at a higher order motor area. And sort of right now, sort of as we look at this image, this is the area that, for me, looks most interesting at this time. This is the area that seems to be specifically activated on the hit trial, and it's almost absent on the mistrial. So there seems to be something interesting going on here at very frontal areas of the brain. And again, all of this precedes licking So here it is in still frames. You'll see that sort of this early sensory response across the first 50 milliseconds or so is basically invariant, and then at later times you see more depolarization across larger areas of the brain, and perhaps in particularly the frontal areas are the ones that look most interesting. And here we now divide hit versus miss trials, and so we sort of take the ratio of these two. We look for the modulation of the sensory response, and here sort of what's labeled as evoked here. So this is the early response of the first 50 milliseconds, the peak of response in the first 50 milliseconds, and you'll see it's almost invariant. It's sitting pretty close to 1. Hit versus miss trials are almost identical. There's perhaps a small modulation here already of frontal cortex. We don't yet know whether that's statistically significant, but what's very clear is that if we go to the late phase, a lot of the brain is more active on a hit trial, but there's a couple of spots that are standing out here, and that's the frontal areas, whisker motor cortex, and sort of more generally this frontal area here that are much stronger in terms of the hit versus miss activity. So there seems to be something interesting going on in frontal cortex that we'd like to investigate in more detail in the future. And once again, I'd like to just sort of draw some analogies with what's been reported in the human EEG literature. This is again a work from Stanisław Dehaene, where, let's see if I can get the, yeah. So we have sort of the, again, it's the same task as before. We have sort of a different masking of visual stimuli, and this is one that perceptually to the human subject is invisible. This is the stimulus that perceptually was reported by the subject, and you'll see again that there's sort of an early invariant response that presumably sort of the stimulus-driven response, and then depending on the percept of the subject, then for the trials that turn out to be invisible, there's not so much activity, but in the seed stimuli, you'll see that there's interesting spread to frontal areas that, at least to some extent, perhaps look a little bit like what we see in the mouse. And so there could be interesting similarities again here between human perception and what we're seeing in the much simpler system of the mouse, this whisker detection task. So what we think we sort of have now sort of seen is that there's clearly a role for, or there could be, there are at least correlates across many cortical areas in the brain. We are relatively sure that S1 is involved and is playing a critical role. So the sensory information, presumably all of it seems to transit through S1, and we think that there's an interesting role for GABAergic inhibition, or rather disinhibition, that allows the sensory percept to be reported, and that could involve a communication between multiple different cortical areas. And in particular, we're quite interested now in frontal areas, motor cortex, and perhaps higher motor areas that could be forming interesting reciprocal loops. And I sort of quite like the idea that there could be some interesting positive feedback interactions between motor cortex S1 and maybe an M2 area over here that are involved jointly in generating the sensory percept, and that we might be able to take out any one of these nodes, and that might have a sort of equivalent behavioral deficits. And of course, that's something that we need to now check on. So we have, I think, correlates that are relatively clear across a large area of the dorsal sensorimotor cortex. And the next question, of course, is whether any of that activity is causal. For S1, we've investigated it. We think that there's causal activity both in the early and the late depolarization, and now we're going to start doing sort of optogenetic experiments where we simply drop light pulses across the dorsal surface, and we'll see what substitutes and what doesn't, and which areas we can inactivate and make a difference to the behavior of the animal. So neocortex, we think, in a sense, perhaps surprising, given how simple the task is playing a prominent role. But of course, we also need to know what neocortex is doing, what the output of neocortex is, because somehow we've got to connect to motor neurons that are actually doing the action. And of course, one possibility is that neocortex simply projects down to brainstem, and I think M1, S1, and most of the neocortex has brainstem projections, so that's, I think, entirely feasible. I think the only area that doesn't project to brainstem is visual cortex, so we can sort of pick more or less any area here, and they could have a profound influence on motor output through sort of relatively direct connections with brainstem motor nuclei. But others, basal ganglia people, including Haggai and others who have been encouraging me for years to sort of go to striatum, have suggested that this is probably quite a likely role for the striatum to be playing in a very simple sort of goal-directed task with reward-based learning. It seems that there could be an interesting role for striatum in this sort of very simple task. And so over the last years, and this has been a collaboration through the sort of the Hebrew University EPFL funding together with Haggai Bergman, we've begun to investigate what's going on in the striatum during this detection task. And there's sort of a number of features that I think are sort of fun just in terms of thinking about striatum and its potential role. And for me, the most interesting one is the potential role of dopamine as a learning signal. I like the hypothesis of Wolfram Schultz that we give a sensory stimulus that's then, in a sense, a reward predictor for the animal. The animal should learn that whisker stimulation is a predictor of reward availability. And so there could be a phasic dopamine signal that comes out and might interact in the striatum and might form an interesting learning signal. So we haven't started looking at learning yet, but we just thought we'd see whether the striatum seems to be playing an interesting role simply as a correlate. And so the work that Tanisipi's been doing, funded by this Hebrew University EPFL collaboration grant then, was to obtain membrane potential recordings from striatal neurons and see whether there are interesting correlates there or not during this detection task. And so this is sort of example hit versus miss recordings from striatal medium spiny neuron. And in this particular recording, I'd say things look fairly similar to what we saw in the neocortex. So we stimulate, there's a sensory response. In this case, there's an action potential being fired. There's some late activity. That late activity precedes licking. And of course, the animal is rewarded on the hit trial. There's another hit trial. There's an early sensory response, some late depolarization, and the animal licks. Miss trials, we stimulate, early sensory response, perhaps less late depolarization, and similarly here. So this is sort of an example that, to my mind, looks pretty much like neocortex, and at first, I must say, I wasn't that excited about it. So as we sort of kept collecting data, however, the picture sort of began to sort of differentiate itself in a sort of more interesting manner, where so we now put the data together from the data set that we have, 30 cells that Tanya reported from, and we see that there's actually a very prominent difference in the early sensory response. So we stimulate, and there's a relatively rapid sensory response here in the striatum that's larger on hit trials than it is in miss trials. Late on, there's a huge difference in activity in the membrane potential of these cells. It's also translated into action potential firing of these neurons. And again, much of this depolarization precedes the licking of the animal.
Is the depolarization precede the one seen in F1?
So the first depolarization here follows S1. So we think most likely striatum receives its main sensory input from cortex. So there is also a thalamic pathway that comes in, and so we can't say for sure where the sensory information is coming from, but there's really a very large projection from S1 to striatum. So this is a major output of neocortex, is the dorsolateral striatum. So it's one of the big targets out of S1. And so we think it's likely that it's coming from S1. So we haven't done these experiments yourself in anesthetized animals. That Silverberg at the Karolinska Institute has done experiments where he's been looking at whisker inputs into striatal neurons, and he finds that all the input is going via S1 under anesthesia. What happens here in the awake behaving, we don't know. And so the statistics then show that there's a difference in the early sensory response and, of course, also in the late component. So now, that's fine, and I think that's interesting. This early component here could then sort of contribute to sort of driving later activity across the brain, and I think that's an interesting possibility. But when we think of the striatum, there's two sort of very obvious different types of projection neurons in the striatum. There's the D1 neurons that are sort of the go pathway. This is the direct pathway where the D1 cells project to substantia nigra, and there's a D2 pathway which projects them to intermediate stations of the globus palatus, and is sort of more or less a stop pathway so we thought, of course, it would be interesting to see if there are any differences there if we can subdivide the cells. And so we have 30 cells in the data set. Many of them are anatomically recovered, and for 5 cells in each group, it turned out that they were identified in terms of the cell types that we were recording from. So we recorded in D1 pre-animals that express TD tomatoes. So these red cells here in the striatum are D1, the direct projection, is sort of the go pathway, and these are then recorded in the D2 pre-animal, and these red cells then here are sort of the indirect pathway, which sort of be the no-go, the stop pathway. And what we were fascinated by was that there seems to be sort of really an obvious difference in the behavior of the D1 versus the D2 cells, where the D1 cells, they had an early sensory response. They all had differences comparing hit versus miss in late periods, but the D1 cells, they had an early sensory response which was basically absent in the D2 cells. They only had this slow, late depolarization that actually correlates with licking behavior of the animal. So this is sort of individual example experiments. This is the 5 cells, so okay, the N numbers are not great, but that's the data set that we have. The 5 cells, if we take the grand average here, you'll see that there's a prominent early sensory response, bigger in hit than in miss trials, and the D2 cells on average just have this slow, late depolarization here. So we think that this early component here in the D2 cells here might be one that contributes to driving the motor output of the animals. And so in data that we're still in the process of analyzing, we've also done now optogenetic experiments where we can stimulate D1 cells, and that substitutes beautifully for the whisker stimulation, and if you stimulate the D2 pathway, that blocks all licking of the animal. So it could be that sort of this early divergence, this early sensory response to the D1 could be involved in driving downstream motor pathways, and of course could affect the rest of the brain in a variety of different ways. So we sort of like this idea that maybe cortex is functioning via striatum, activates D1 pathway in a sort of rapid go signal, might be inhibiting substantia nigra, that in itself then might allow release of activity in brainstem motor nuclei, so there could be sort of relatively direct motor pathway, or there could of course be sort of a slightly longer loop via the thalamus that then might be involved also in the generation of the late depolarization that we're seeing across rather widespread regions of the neocortex. And so of course the critical test that we need to make is inactivation experiments in striatum to see whether there's actually a causal role for that activity, and that's a more difficult experiment, and we haven't yet managed to get that to work. So that's where we are right now in this detection task. We, of course, still have a very long way to go before we really complete a sensorimotor loop, but at least I think you can see that we're beginning to try and put some pieces of the jigsaw puzzle together, and we hope that sometime over the next years we'll sort of have this sort of real quantitative detail, say causally what the different steps are, how the sensory information is bouncing around the brain, and how the animal performs the task. And then, of course, we'd like to know how the animal learns that, and that will be a whole other ball game. So I'd like to finish then by thanking the people who did the work. So Shankar Satchidanandan did all the wholesale recordings from the detecting animals. Rune Srinivasan did most of the optogenetic substitution experiments. Tanya Sippy did all the striatal membrane potential recordings. Alexandros Kyriakis from the voltage central dye imaging. Damien Lepre and Sylvain Crochet are involved in the optogenetic striatal experiments. And I'd like to thank you for your attention.
First, thank you. Great result. Still, the late response don't come back even after 1 second. What is the time course of this? In your recording?
Yeah.
But in the striatum, but also in the cortex. It continue more than 1 second.
Right. Well, so what I think I've shown you I've shown you some. Okay, there's nothing here. So the licking of the animal often persists for quite a long time, and so we think that there could be sort of motor components that are simply just involved. The animal's licking, and it gets a droplet of water, but it keeps licking for some prolonged period of time. Now in the striatum, the situation seems to be a bit more complicated, and there, I mean, here we really have extremely long depolarizations, much longer than we see in cortex actually. I mean, that is still a bit of a mystery to us. I mean, in the end, of course, it does come back. So it's not that we get always depolarizations, but it is extremely long-lasting, and we're not sure where that's coming from. And sort of, I think one potentially interesting thing is whether there are then additional things going on that might be interesting, whether dopamine is being released and causing some other interesting interactions that but we haven't started studying that yet. But there is something, especially in the striatum, unusual about the membrane potential responses that are really much longer lasting than we anticipated.
You see this depolarization during the post-clown trials? Depolarization, the same depolarization that you see during the heat trials. You see this during the post-clown alarm trials?
Yeah, so in the neocortical recordings, I think we have too little data there, and so we haven't analyzed that carefully. I think it's a very good point. We ought to. In the striatum, we've been looking at it in some more detail, and false alarm is for sure associated with depolarization, but again proceeds with a very similar time course actually to the depolarization driven by sensory stimulus. So there seems to be something interesting there, and it's almost equivalent in amplitude. It's a little bit bigger, the sensory evoked for the false alarm. It's almost equivalent in terms of if you see a line to first lick, then there's within the 200 milliseconds of before, it's a big depolarization.
Now, these mice, they sit there and they learn the task, and we still don't know about the learning stage, but still, you could, did you look at the activity before the onset of the stimulus, in the beginning, at the end, or after a failure, after a miss, etc., that would change the person? So this activity that comes there before, the -200 or so.
Right, yeah. So we have been looking, I'd say, and we've never found anything yet, unfortunately, so neither cortex nor striatum.
Probably depends on the design, because especially if you averaged or you did select trials by the previous results or something like this.
So, I mean, here we simply select on whether the animal licked or not. And here we don't make any selection, and we simply look, and we found, I mean, the sort of the mean membrane potential is not different. We looked at sort of the possibility, we were hoping that there would be changes in what we sort of think of brain states in terms of looking at oscillatory sort of FFT activity, no difference. So that's been disappointing, I would say. And my guess is that there probably are simply multiple different failure modes. My guess is that if we do things, make the test perhaps more difficult, maybe we start reducing stimulus strength, then we might see sort of things where brain states and other things might make more prominent impact upon it.
Or just sorting this activity by the previous results, by the previous trials, I mean.
That we haven't tried looking for, so that we don't know. So we haven't looked at sort of inter-trial dependence and whether you sort of get into interesting interactions across sort of but we've certainly been looking for pre-stimulus, because I think in a sense that would be the nicest thing to figure out, is that there must be some signal somewhere in the brain that's different on the trial, which turns into a hit. And well, it could also just be random, but okay, we don't like random so much, so we expect that there is we expect that somewhere in the brain there's something, but it's not obvious in cortex, it's not there in striatum either as far as we can tell, and so yeah, we keep looking, but we haven't found it yet.
Okay, one last question.
You showed quite convincing evidence that this late response is something that correlates with the hit or miss, or with the formation of the correct first sector. This is only in the context of a behavioral task. You'd imagine that formation of a first step doesn't depend on behavioral tasks. What happens in an animal that you're not trained?
Right, so I mean, I think sort of perception, of course, experimentally, we don't know about perception if there's no task. So okay, we simply don't know. So I think we can't really ask that question. But of course we can see what happens in naive animals, and sort of basically, and I don't think any of the data is on any of these slides. No, I think that's just out. But the sort of, I say sort of the, just so we sort of take this, I say the control data from naive animals looks quite a lot like mistrials, I'd say. So the late component is that's missing, and we have, if anything, the sensory response is larger in naive animals, and we think that's because of a brain state change. So brain states do make a huge difference to sensory processing. It's just that in our particular experimental conditions, we don't get a big variety in brain states. So it seems that the animals adopt one brain state that they seem to keep, at least for as long as our recordings last, which, it's not as long as we'd like them to be. So of course, if we had longer duration recordings, we'd probably see some more scatter in the brain states. So basically, it looks like a mistrial. So this late component is missing, and so depends then on how you want to sort of interpret perception. And I think probably the better way of thinking about this, as I say, although I've used the word perception quite a bit, is really as a sensorimotor loop. And I think perception, that's a big word. But we do require a motor output for perception experimentally. And now, of course, yourself subjectively, internally, you can have whatever percept you want, but I will never know about it if you don't tell me. So we do need a motor output if we want to consider perception in any setting and gather data on it.
Okay, so